Hi, everybody. We'll wait just a minute or so to let folks join before we get started. And while we wait, I am going to put up some QR codes. Hopefully you can see that. Some QR codes for our mailing list in case you found yourself here, a forwarded email or something. And then also a new form for speaker nomination. We're trying to expand our network and invite list a little bit. If you're coming on in, we're just going to wait just a little bit longer to let folks join. All right, I think we can go ahead and get started. Today we have two great talks coming up. First up is Fangqiong Ling from WashU St. Louis. Very excited to hear about some of her work she's been doing on WBE since I think even before COVID a little bit. And I have the wrong name there. I apologize. I whipped up these slides really quick. The second talk is not is she again? The second talk is Aaron Bivins from LSU, and he's talking about passive sampling to scale environmental surveillance. So sorry about that, Aaron. Next month, well, we have one committed speaker, Tyson Graber from the Children's Hospital Eastern Ontario Research Unit. He's going to be talking about host response, sort of immune response through wastewater. And we're still finalizing our second speaker. So I'll put up these QR codes again if you need to register for the email list. But please also, if you have ideas for different speakers, I know there's a lot of work going on in this area. It's not possible we can know everyone, and don't be afraid to self-nominate as well. We're looking to expand our network as much as possible. I'll also put the links in the chat if you'd prefer that. So with that, we'll start off. Fangqiong, you could go ahead and share your screen and get started whenever you're ready.
You're muted.
Perfect. Okay, thanks, Ali. Thanks, Cal, for the kind introduction. And so today I'm going to share a few studies that our group has been working on about WBE. So the main one of the main themes in our group is trying to dig out the information from microbes in urban water infrastructure and use it to learn something about the infrastructure status, and now with WBE to learn something about the health status. And with this group, I do not need to go into details about how amazing WB has been and will be. The main idea is that WB offers a new method for collecting information on health status in an urban environment. And so there are many benefits. It help reduce reporting bias, help reducing test fatigue, and it's a low cost and actionable method. And beyond the COVID-19, there are a wide variety of pathogens and their variants that can now be detected by WB, thanks to the amazing work from the researchers in this field. I'm sure some of them are in this webinar as participants. And the first study I want to introduce is a meta-analysis study. So why do we want to use the meta-analysis methodology to look at WBE data? So there is a saying in that I read about meta-analysis. that is important questions asked many times at many places. And this is so true for WBE. Ever since the start of COVID-19 pandemic, there's a lot of research and resources that has gone into wastewater-based monitoring. And meta-analysis, there are many definitions on like what exactly goes into this term. And the definition that I really liked is meta-analysis is a research process used to synthesize systematically and merge the findings of multiple independent studies. And there are procedures that are available to quantify effects. And meta-analysis is useful in many ways. And something that I find very helpful is it can help us move beyond statistical significance and help us look at magnitude, direction, and variance in effect. And a single study is usually we can only one research group or one research team has only so much effort, so much time and resource to generate data. And when we use meta-analysis, a major benefit is that we can generate citable statistics from a large sample size. And this procedure has been found useful in evidence-based healthcare. And so one analogy that I really enjoyed is meta-analysis help us to look at the shape of the forest while still retaining the information about the trees. So this meta-analysis work was conducted in 2021 when we were a year into the COVID-19 pandemic. And we asked several simple questions. One is when we pull all the report about SARS-CoV-2 WBE together, what is the overall proportion of positive detection? And this is a question brought up by our amazing postdoc scholar, David Mantilla. And David has performed monitoring of SARS-CoV-2 using wastewater. And something that he found is that he doesn't always get a positive, even though we were in the middle of pandemic. And the next question is that what's the overall viral RNA levels of SARS-CoV-2 virus in wastewater collectively? And the third question is what's the overall strength of correlation between positive detection of the SARS-CoV-2 RNA levels, the SARS-CoV-2 RNA or its levels in wastewater to that of existing epidemiology indicators, for example, daily or cumulative cases. So one step that we require before conducting meta-analysis is to perform a systematic review. So this process helps us define what studies are included in the meta-analysis, basically help us define the population. And so in our study, we used the Web of Science core collection, and we conducted a systematic review for literature published up until February 16th, 2021. And we used a simple keyword, SARS-CoV-2 and wastewater or sewage. And this procedure generates 101 paper at a time. And now if you do this search, that's going to generate orders of magnitudes more studies. So after we collected this set of papers, we set a few criterion to help us generate the data set from these studies. So we look for studies that have reported qPCR data and following a few units. And then we look at the sampling locations of the studies. So we look at sampling locations such as raw sewage at wastewater treatment plants, sewage collection network, lift station, manholes, or septic tanks. So this criterion seems very obvious now, but at the start of the pandemic, the studies about SARS-CoV-2 in aquatic environments were very, very diverse. So for example, there are studies about effluent and understanding the risk that effluent may pose to the aquatic environment. So we want to focus on the raw sewage aspect. And the third criterion is that SARS-CoV-2 COVID-19 case records were also reported. So this is because we would like to seek correlation between the case records and and the wastewater SARS-CoV-2 RNA levels. And we believe the authors are the best informed as to what kind of case records are most representative for their studies. And so after this procedure, the studies were trimmed down to 20 studies, and that gave us 1,751 observation and 671 unique samples. So we went further on with this systematic review procedure. So something the systematic review start to inform us is that there is a diversity of methods that are used to generate the SARS-CoV-2 RNA readings from wastewater. And we performed so in order to have data that are more or less using similar methods, we performed a subgrouping. So one subgrouping step is at the collection method. So here we saw that there are studies using grab samples or composite samples. And this is important because the grab sample and the composite sample may represent different population sizes. And another aspect is which fraction was analyzed. At the start of pandemic, there's many methods used to concentrate or not directly analyzing the SARS-CoV-2 virus from wastewater. So some studies choose to use the supernatant, and some studies choose to look at the pellets, and this will involve different assumptions about where do we detect the virus more. So we put a subgrouping on this aspect as well. And so overall, we look at the positivity of these observations, and we found that the overall positivity is 0.68, and it has a 95% confidence interval of 0.52 to 0.85. So this is very encouraging. So we're very so the overall positive detection confidence interval is above 0. So we're still very confident we're going to detect something from this wastewater. And the overall positivity actually depended on the sampling mode and the fraction analyzed. And we see a higher detection rate from the composite samples and from samples that involve solids. And a similar trend was detected when we analyzed the SARS-CoV-2 RNA levels. And here we also see that the solids or the mixture of supernatant and solids generate a higher level of SARS-CoV-2 RNA. And so when we put these results together, the mixed effects model gives us a very powerful framework to synthesize the data. So we look at the correlation between SARS-CoV-2 levels or positive detection against daily cases or cumulative cases. So here's the results about daily cases. So what we detect is that overall we have a positive correlation. The coefficients here, they are positive. So that's great. So that means when there are more cases in the urban area, that's associated with higher detection rate or higher SARS-CoV-2 RNA levels in the wastewater. And but when we look at the R-square, we start to see that if we only focus on the cases alone, the ability that we have to explain the variation in the positive detection or the RNA levels are pretty low. So the adjusted R-square is about 0.09 or 0.009. And one very powerful aspect of the mixed effects model is that we can incorporate the study-to-study variations. And once we involve that, the R-square is much higher. So this means a lot of unexplained variants are covered in this study-to-study differences. So what do we learn from this analysis? So first, still we see a positive correlation between SARS-CoV-2 RNA levels and current epidemiology indicators. So this is reinforcing the validity of WBE in COVID-19 monitoring. And we saw that composite sampling and solid fraction associated with higher positivity, RNA levels, and strength of correlations. And that's potentially useful for other new applications. Another aspect is there's a large heterogeneity between studies, and that's partly explained by methodological variables. And we also saw system-to-system variations. And this means there is a lot of potential for method development focusing on increasing reproducibility, systematic documentation of the data. So this paper was published in 2022, and a lot more studies have come out to look at the detailed mechanistic and methodological aspects. And this reference from Professor Lawrence Steller's group, I found that it's telling us something very informative. So this study shows that when the methodology is expanded to more targets, there's no single method that's optimal for all. And a similar observation that solids method is helpful. It was observed in this study. And another aspect about open and systematic documentation of data was also brought up by a broad group of researchers. So standardizing data reporting in the SARS-CoV-2 wastewater surveillance seemed to be an idea that has been supported by many researchers. So I mentioned briefly that if we look for wastewater-based epidemiology research today, the amount of research has really grown dramatically, and so this motivated us to think about are there ways that we can use GPT models or other large language models to help us facilitate making the literature synthesis faster. And so we used this meta-analysis as our data set that has been validated by human eyes, and we look at how well does the GPT models detect whether there is actual original data from the paper and whether the sampling location is identified as a wastewater treatment plant, lift station, manhole, septic tank near a building. And we tested a few models, and we tested different inputs to the model. And we look at the performance of the model by precision, recall, F1 score, and accuracy. And among these metrics, we put more attention on recall because we anticipate this machine-assisted literature screening to happen in the early stage of systematic review. And so we want the process to be more inclusive. And from this study, we found that so for the first question, the GPT models actually performed very well, and models with or without fine-tuning were both able to achieve a very high performance with abstracts as input. And as to the second question that's more specific about sampling location, we see that its performance was the models that we tested has moderate performance with abstract, but once we use methods as input, the recall can be improved and go up to 92%. And fine-tuning step did enhance the performance. So I really like a quote that's from our student Kasaba Chibri. So she said she really liked this study because it shows that these GPT models, it can be validated as a tool instead of being used as a shortcut. And I also want to add a little note that still from what we see, the models at its current stage cannot do everything in this systematic review process. Okay, so I guess with time, this is maybe a good place to open up the discussion. And I want to briefly mention that our group has another another wastewater-based study that focus on tools to extract information from a wastewater sample and focus on extracting the human population size from a wastewater sample. And I'm happy to discuss that's the aspect offline. And I want to thank our our amazing students and our funding sources, and the wastewater-based work is supported by an SF grant under the CBAT division, and also a faculty startup grant from the McKelvey School of Engineering. Thank you.
Great. Thank you, Fangqiong. Just as a reminder for folks, any questions you have, go ahead and put that in the Q&A box at the bottom of your screen. I have a question from Allie that had to come through the chat, so folks don't see it, but I'm going to put my own addition on it. So Allie asked, before you presented the LLM models, if you were planning on re-evaluating your meta-analysis with all the new papers that continually come out, or include other new targets, for example, influenza. And I'll sort of add my own spin on that. Is that something that you think could be almost automated into an LLM as the literature continues to grow and grow? So those two aspects there.
Yeah, those are great questions. We've been thinking about this for a long time. So I think with the amount of literature that we have right now, having some kind of large language model to facilitate it will definitely be very helpful. So colleagues at EC are looking at using LLM to facilitate the synthetic biology research, and there are a lot of commonalities in the kind of text that's available in these biological procedures. So yes, I think there is definitely space to incorporate language models in facilitating literature research synthesis. And my advocacy would be that we still provide some kind of human validation, because so it still this at least from our preliminary study. So sometimes the data extraction from the models themselves, they do not always extract the right text. So I feel that like validation in this machine-based literature synthesis is really crucial.
How do you think that's going to change as these models continue to be updated and apparently improved? I have not dove as deep as you have on it, but it seems like every few months there's a new sort of supposed improvement. Do you think that will become less and less true, or what has your experience been with applying these tools as they improve, GPT-3, 4, or 5?
I'm not an expert on large language model, but just from our experience, so sometimes we kind of need to think about ways to help the model be more careful. So like, for example, focusing on some part of the text, or like do not just look at one part of the text. So that kind of procedure, at least in my current experience, that still helps. Yeah.
All right, a few questions from the Q&A. Yuki Wang asked, can you talk about the cost, the API cost for LLMs for the literature review, and which model performed best in your study?
Oh yeah, so in the paper that we published on environmental gold, that we recorded the cost. So the fine-tuning cost is about at the dollar scale. So it's a very low cost. Yeah, much lower than a minimum wage.
Yeah, and which model performed best?
At the time we tested the GPT-4 models, that was before GPT-5, so GPT-4 models did perform better. Yeah.
Great. Another question, do you have any recommendations for future modeling studies to enhance WBE accuracy?
I'm going to do a little bit promotion. Can I share a little about our slides?
Yeah, of course.
Yeah, so this is something that I wasn't sure that whether we had time to present, but yeah, now we are asked this question. So I think something very unique about WBE is that it's a kind of sample about a population, but the population size or the sample size in this case, the sample size is not always known. So if we take a wastewater sample from the sewage collection system, the sample size that we really care about is how many human population, how many humans feces was captured by that sample, and that is not something that's as easily known as other kinds of experiment. For example, if I do an experiment in a lab, I always do know like how many replicates I did, and that's my sample size. And if I'm testing if I'm testing SARS-CoV-2 from, for example, nasal swab, I always know how many participants send in their nasal swab and how many swab turn out positive. So then the total number of swabs coming in would be the sample size, and then the positive swab can help us. Combining these two numbers can help us know the positive detection rate. But in the case of wastewater-based epidemiology, we do not always know the sample size. And I think that's a very unique aspect about this kind of data. And so our group approached this problem from a microbiome perspective. And there's a publication on PLOS computational biology that we look at. We provided two perspectives about estimating the human population size from wastewater. One perspective is to use CCNS ribosome RNA gene sequencing and look at the relative abundance. Another perspective is to use metagenomic sequencing and look at the within species diversity of gut-associated bacteria. And I'm sure there are other ideas, and I think that's an area I'm really excited about.
Awesome. Glad you're able to share that in a meaningful way. So
Thank you, Cal.
that's great. All right, I think we have two more questions. Can you explain between study heterogeneity by modeling treatment plant characteristics? So percent of industrial effluent, for example, population size. Did you look at that with your study as well?
I think that's definitely something that will be very interesting to look at. So in our study, we didn't use a modeling approach to look at that. But if we are going to do a meta-analysis today, I would definitely want to include that because now we have this large amount of literature. So at the time that we performed the meta-analysis, eventually only 20 studies met all the criteria, and we put that as a discussion aspect in the meta-analysis paper. And there are many interesting aspects if we think about how complex wastewater is, right? There is combined sewage and there is a separate sewage, and there are there are wastewater treatment plants that are taking in the industrial wastewater, and there are treatment plants that do not. So I think the complexity of wastewater collection system is amazing, and this data-driven method will help us learn more about how the complexity affect the WB data.
Great. And then the last question also came from Allie. Did you find that the authors are providing their data and their papers in a format that allows you to extract it and use it in the meta-analysis? And do you have recommendations for people on how they should make their data available?
Well, I appreciate the question. So actually, the meta-analysis, it looks like a modeling work, but the data generation part is really hard work. So our authors, I really appreciate like David and other authors on this paper, our amazing students. At the time we performed this study, our postdoc and students really went into the papers and find the numbers. And sometimes we need to extract the data from the figures. And we were like thinking, oh, it would be amazing if these studies they come with a spreadsheet as their supplementary, and if all studies come with a spreadsheet, and the kind of like minimal information standard that's advocated by the ES&T editorial, I think that's also going to help tremendously. So some kind of systematic format for depositing WB data that will be tremendously helpful.
Great. Thank you, Fangqiong. And if anyone comes up with additional questions, I'm sure she'd be glad to answer, have a discussion over email as well.
Yes.
So with that, well, I'll introduce Aaron Bivins. Apologies again for not updating the slides sufficiently, but Aaron, you could go ahead and share your slides and get started whenever you're ready.
Aaron, you're also muted.
Batting 1,000.
That's
All right.
better.
Thanks for having me, everyone. I'm going to share some of our work on adsorption-based sampling of microbes from water. Um, Here we go. So my name's Aaron Bivins. I'm at LSU. I lead the micro to macro lab. Our goal is to integrate microbiology into macro level decision making. We really do this in kind of a chain of events. We develop new ways of observing microorganisms in the environment. We build models using environmental microbiology data to translate into public health impact. And then we use our models to test policy and design relevant hypotheses. And the goal is to ultimately integrate environmental microbiology into policy and design through computational modeling. The work I'm going to share today was led by Shimul Ghosh. He's a PhD student here at LSU. Malik Smati, who was an undergraduate researcher we hosted for a summer. Ronnie Carter is our kind of local wastewater guru. He's here in the middle here with the New Orleans Saints hat. He's our fixer. So the plant we worked in, Ronnie helped us out to get access. And then, of course, Emily Hayes and Graham Gaillon from Dalhousie University. They published a lot of the early work on GAC and were nice enough to come and visit and show us the ropes. And now we're kind of trying to build off the foundation that they laid. So I want to start with kind of this data risk paradox. And this is something we face in WBE, but we face it in a lot of ways, a lot of different aspects of environmental microbiology and human health. And that's the idea that the data is most scarce where the risks are greatest. This map is kind of old and probably not up to date, but what I want to point out here is the bright green dots are the wastewater plants participating in news at the time. And then the dark maroon counties and parishes, as we have here in Louisiana, are in the upper quartile for social vulnerability. And we can see that the green dots don't really penetrate these maroon spaces very much. And so one of the reasons for that is might be the resource intensity of the methodology, right? So um, composite sampling with an auto sampler is the gold standard for wastewater surveillance. You can see on the left, this is our setup at when I was at Notre Dame. Probably like 20, 25 grand to put this in, run the power, just a lot of requirements to set one of these up. And so that drove a lot of interest in low cost, resource efficient methods such as passive sampling shown on the right. In this case, we have a tampon swab tied off to some fishing line, clipped into a manhole and left in place. And, something like 250 to deploy one of these if you can get access to the manhole. And so this idea of passive sampling, although it enjoyed kind of a renaissance during the pandemic as people deployed them, it's not a new idea. As early as 1916, some scientists put sterile cheesecloth onto wires and deployed it out into a stream for a few hours to look for tuberculosis downstream from a health resort. And then it was really in 1948 with the invention of the Moore swab by Brendan Moore that passive sampling really kind of became like a global phenomenon. The Moore swab is, a piece of gauze that's been folded over itself a few times and then tied off with a string. And Dr. Moore and his team became extremely proficient at tracing through sewer systems. You can see a map on the right from one of their papers and identifying salmonella carriers in towns. And they found that the Moore swab gave them increased sensitivity over what they called catch sampling, or we would call grab sampling now. So what makes passive sampling work? The operating principle is adsorption. And so adsorption is a mass transfer process where an adsorbate, in this case, Salmonella typhi or SARS-CoV-2, from the sampled medium is transferred from the sampled medium to the adsorbent by a free energy gradient. And so what's great about this process is under certain conditions, it's thermodynamically spontaneous, which means we don't need any external driving force. So we simply immerse our sampler into the fluid, and this process drives the rest. We know quite a lot about microbial absorption from a theoretical perspective, right? We know that it's a function of the interaction between the surfaces of the bacteria or the virus and the surfaces of our sampler. What's challenging though is that these surface features, how they interact is a function of the characteristics of the medium in which they're immersed. And so in the case of wastewater, right, these are characteristics: temperature, ionic strength, viscosity, hydrodynamics, pH. These things change in time and space. And so it creates a very complex system with several different mechanisms that are in play all at the same time. And so our interest on my team is, can we take this theoretical framework and employ it to develop what we call quantitative passive sampling, which is the idea that is shown in the right panel. We take some adsorbate, GAC, we deploy it into the water column and absorb microorganisms over some exposure duration. We then recover the adsorbate, enumerate the microorganisms, and the question is, can we take what we know about the theoretical frames and back calculate a microbial concentration in the water column based on a predictive understanding of adsorption? From a historical perspective, passive sampling really hasn't been quantitative. There are 155 publications since 1916, 156 now. There was just a new one last week. None of them take what was recovered from the passive sampler and quantify what's in the water column. You can see all the gold circles. Those are all the SARS-CoV-2 passive sampling papers. So that's the renaissance I was talking about. There are two excellent reviews in Water Research that provide a nice overview of the field if you're interested. For SARS-CoV-2, of course, people started deploying a lot of these more swabs again, especially at universities trying to surveil dormitories. In an earlier paper, we aggregated data from a couple different settings. And what's interesting is that our results imply that maybe under certain cases in certain settings, there is a quantitative relationship between what's measured by composite sample here on the x-axis versus what's recovered from a passive sampler here on the y-axis. You can see University of Maine an R-squared value of about 0.76. At the same time, though, that's not always the case in all settings. You can see Yukon, the R-squared value is 0.27, and then overall, we're sitting at about 0.41. So these results imply that in some cases, there is a quantitative relationship, and in others, there's a weaker relationship between what's measured by these two different methodologies. These are with more swabs. So one material that Emily Hayes and Graham Guillon started working with is GAC, and empirically, GAC is actually quite promising. These are two kinetic models. I think pepper-mild model virus on the right and crassphage on, pepper-mild model virus on the left, crassphage on the right. You have time on the x-axis versus the amount adsorbed onto the sampler on your y-axis. And you can see the R-squared values in these batch reactors. So this is lab scale batch reactors are actually quite high. Our team's interest was, okay, in a batch reactor, what you do is you measure in the water column, and any analyte that has disappeared from the water column, you assume is on your sampler. But we really don't have that luxury when we deploy these in situ. And so our study design was to look at the performance, the quantitative performance, based on what we actually recover from the samplers, using an in situ study design. And so over 90 days, the summer of 2023, we conducted 19 deployments with durations from 40 hours up to 500 hours. Our sampling car broke down, and it took us a few weeks to get it fixed. And this site's about an hour and a half from campus, so 3 hours round trip. So we didn't do any deployments less than 40 hours. We ended up with 38 unique samplers. We eluted off the sampler with a buffer, extracted the total nucleic acids, and then we performed analysis with digital PCR, and then we did some 16S amplicon sequencing. So viruses have been kind of the big players in passive sampling more recently. So starting with adenovirus, you can see that we recovered it. The blue line is the sampler in front of screen 1. The purple line is in front of screen 2. We consistently recovered adenovirus. You can see so the length of the bar is the deployment duration. You can see our 503-hour deployment here in the middle. We see this uptick in adenovirus late in the study period, despite the fact that the caseload was quite low on a state level. What's interesting, I don't have it on this graph, but in the month after our study concluded, classes started, so we had to suspend our sampling. We saw a large uptick in the adenovirus positivity in the state of Louisiana. And so maybe our, small plant serving 4,000 people in rural Louisiana is a sort of an early indicator here. For SARS-CoV-2 and RSV, this was over a summer period when positivity started quite low, actually. So we have a regional positivity for SARS-CoV-2, state positivity for RSV, and really just sporadic detections until later in the summer when we started picking up SARS-CoV-2 RNA pretty consistently off of our samplers. The reality is the data really precluded a more robust quantitative analysis, but there seems to be some alignment in these trends. We also wanted to extend the footprint to bacteria, so we looked at MAP-A, which is an indicator, a target associated with Campylobacter jejuni. We recovered it pretty consistently early in the study period, and then our samplers went to non-detect throughout the back half of the study. And there's not really clinical data for us to compare with on a regional or state basis. We took some of the nucleic acids and we did 16S amplicon sequencing. These are bacterial families on the y-axis here as a function of exposure duration. We were interested in, is the community composition consistent? And with the exception of Rhodocycliaceae here, you can see our relative abundance started quite high at 44 hours and then decreased throughout as the exposure duration increased. But in terms of our sewage-enriched families, our biofilm-associated families, and our fecal bacterial families, we don't really see large shifts in community composition, at least in this data set. For ARGs, we also looked at a couple of ARGs. Again, exposure duration on the x-axis. We were worried about enriching ARGs at some point. And what we saw is that at least over 168 hours, we didn't see a noticeable temporal trend in terms of the abundance of ARGs recovered from the samplers. We did this for relative abundance too, and the trend is the same, although I haven't shown it here. Probably the most interesting part of this is the kinetic performance. So what I've got on these plots is the sampling rate on a log scale per hour versus exposure duration. And what we found was that the average sampling rate over the deployment decreased exponentially as a function of the exposure duration. And this was consistent across 3 different analytes: pepper mild model virus on the left, adenovirus in the center, and 16S rRNA. You can see the R-squared values for PMMOV and 16S are pretty high. Adenovirus in the middle, the R-squared is not very good, and that's our hypothesis is right. The initial adsorption rate is quite high for adenovirus compared to PMMOV and 16S. So you can see we don't have any data inside of 48 hours, and that creates a lot of ambiguity in the early period of this model. But our assertion from these figures is that despite the fact that this is 38 different deployments, despite the variation in the composition of the wastewater that we might expect in raw influent as a function of time, we still see a consistent mathematical model of the sampling rate. Now, what's interesting is that exponential decay of the sampling rate is consistent with a theoretical adsorption model called the Elovich kinetic model. To kind of, I guess, ground truth this a little bit, we pulled the data out of the Hayes et al. Papers, and we did a nonlinear fit on their batch data for PMMOV and crass phage. And what we found was that if you do a nonlinear fit, actually the Elovich model provides a higher R-squared value compared to the pseudo second order model originally proposed. So there's more work to do here to investigate this. This was recently accepted into water research, so if you want to, read all the details, you can look at this QR code. Now our interest is kind of, okay, what else beyond WBE, right? How else can we deploy these passive samplers? And so we're interested in surface water systems. We've done an experiment at the Notre Dame Linked Experimental Ecosystem Facility, or LEAF. There are these experimental streams there where we can actually inject a contaminant plume. We can deploy our samplers, and we can kind of analyze their performance in a very well characterized hydraulic system. And so we did a manure plume release where we injected a cow manure plume. We deployed our samplers along 10-meter intervals down the stream. And we put 3 samplers at each location, and basically we pulled one out at 90 minutes, one at 120, and one at 180 minutes. We know exactly what we injected. We've also paired this with water column sampling, and we're starting to parse this data. One of the things we looked at was TET-W and ARG. You can see in the water column, 2.4 log at 10 meters down to like 2.2 log at 50 meters. What's interesting is we don't really see a large difference in these time, or a large and consistent difference between the exposure time for these samplers. We also expected to see kind of a log linear decrease in the amount on our samplers, and we don't see that either, which is interesting. And we're continuing to explore this data. We also looked at cow M3 and rum tobacco, which are microbial source tracking markers. So cow M3 here in the center. You've got what's in the water, the purple dots, so maybe around like 0.1 gene copies per mil for cow M3. And you can see the prevalence on our samplers decreased as a function of the abundance in the water column. So this is 4 out of 26 samplers that were positive. When we look at rum tobacco, another source tracking marker, a little bit higher abundance, right? 1 gene copy per mil. And we see the prevalence increase on our samplers. The other thing that's really interesting in this system is that we see our samplers are enriching our analyte, meaning that we're getting more than a 1-to-1 mil to sampler ratio, which is promising. Other applications that we're working on now, eDNA. So you're trying to deploy these for eDNA surveillance for rare species. And then we've just got some pilot funding to start deploying these for Vibrio surveillance in shellfish harvesting waters here in coastal Louisiana. So some of the key takeaways so far, the empirical evidence suggests that passive sampling could produce quantitative data. And the GAC performance is especially promising. It was mathematically consistent with an existing absorption model across 38 different deployments in wastewater. There are many other interesting applications beyond WBE where passive sampling could increase our ability to really increase our spatiotemporal resolution for our observations in complex systems. But we need fundamental experiments to further explore the relationship between the driving variables and the adsorption kinetics and equilibria. So that's what we're working towards now. Thanks again for having me, and I'm happy to take any questions.
Thank you so much, Aaron, for that presentation. I don't think we've had a passive sampling talk at the RCN in the past, I don't think. But so it's really great to have you here to talk about it because it is gaining in popularity. If anyone has questions, please put them in the Q&A, and I have some questions here to ask you from Kyle, and then I have some for myself, but I will also take some from the Q&A. So the first question that we have here is that it looks like the sorption is maximized really quickly for the GAC in the batch reactor. Does this imply that what you would detect on this GAC as a passive sampler will reflect what's in the water column over a short period of time, or like that you should only put them in quickly for like an hour or whatever that equilibrium time was? Or what does that say? What does that tell us about the sampling?
Yeah, so what's interesting about those batch reactors is the reason they plateau is because the GAC had absorbed everything.
Mhm.
They seeded a certain amount of virus and all of it had disappeared from the water column. So the question is that saturation or is that just consumption of the adsorbate? You would need to do more experiments with different amounts of the virus. What we saw in our in situ data is that the sampling rate decreases exponentially, but interestingly, it didn't go to zero. It slowed down quite a lot. And my hypothesis is this kind of a multi-layered adsorption phenomena. So your active sites of your GAC get consumed, but then those new layers of material continue to adsorb, but just not as quickly. Um, I think there's still definitely more work to do to come up with like what's that kinetic region and how does that vary as a function of the water that you're deploying the sampler into. Right now, in all these passive sampling studies, none of them are really making like water column measurements of the physicochemical conditions in the water column. And that really precludes our ability to say to start to untangle what's driving this adsorptive behavior. So,
Mhm.
a lot more work to do, I think.
Yeah. Okay, we got a lot of questions in the Q&A, and you guys can see my dog Hunter behind me. He thinks he's a cat. So I one of the attendees asked, does it matter how you get the sample off the swab? Like if you squeeze it or grind it, etc. Like is that part of the method impacting the measurements that you make?
I definitely think that it does. Um, if you look at that's my hypothesis, right? I haven't like done the experiments to tinker with that, but we've messed around with different elution buffers. We've actually so that in that original wastewater paper, we used the buffer from Hayes et al. In that manure release experiment at Notre Dame, we kind of tinkered and made our own buffer. And we saw about a 5 times, we increased the amount of 16S we were getting off by about 5 times. So definitely that's going to be a variable. The other thing too, though, is even sampler to sampler, we saw, 3 samplers at the same point where all of a sudden one of them we had like a log higher bacterial recovery. I think that's an artifact of the recovery, but I don't know how to explain that yet. But I do think that's something we're going to have to contend with. One of the things that we found was that our process control, we seeded a process control during that wastewater study. We seeded SCADA 22 as an extraction control, and it didn't really explain the variation that we were seeing. So controls are going to be an important part of this and a difficult part of this because how do you get a control onto the GAC and then how do you get it off and how do you assess that are open questions.
Yeah. I'll ask one of my questions, but there's still more from the audience too. But I have one question, which is, I know that these passive samplers have been used a lot in the chemical space, like sampling for PCBs and PFAS and things in the environment. And I was wondering if there's like analogies that help explain what's happening, or if there's like a fundamental difference that precludes those studies from informing this work that you're doing.
I definitely think there are lessons we can learn from what's been done in the chemical space. There's been a lot more. Um, I think there's some things we can abstract, and there's other things I think that if we abstract them could be potentially a problem for us. In the chemical space, right, you'll see people kind of try to bifurcate into like kinetic versus equilibrium. And they'll say, okay, as long as you deploy less than 24 hours, you're in the kinetic region. And so you can interpret this as a time-weighted average. In the case of a chemical constituent where the structure is well known, the behavior is a bit well known, maybe that's possible, but you'll kind of see people start to kind of fit linear models to the data, right? I see that a lot in the microbial world. Okay, we just fit a straight line to this adsorption data, even though we know that adsorption really isn't a linear phenomena. Um, so I think we have to be careful with our simplifying assumptions. The other challenge is, of course, these bacteria, their surfaces, and viruses, there's a lot of heterogeneity in their surface kind of features and how those features interact with the sampler. So in that regard, I think it's going to be a bit more difficult compared to like a molecule of some chemical compound. The
Mhm.
chemical approach has been to kind of compound by compound validate your model. I think it would be much more interesting if we could kind of a priori have some idea about the performance based on, you know, the adsorbate and the sampling medium, and that's kind of what we're hoping to start teasing apart.
Cool, thanks. There's a question about the Vibrio work. Is that program comparing involving sentinel bivalve sampling as well to compare with the passive sampling?
So we don't have any bivalve sampling yet, given the budget constraints. It's a pilot project. We are working with oystermen here in Louisiana. So hopefully maybe it'll eventually get to that. But for now, we're just kind of, this is like proof of concept, right? Can we put a passive sampler in place? Can we get Vibrio off of it? Is there a quantitative relationship between what we get off and what's in the water column? Those are kind of our initial questions.
Thanks. Okay, and then we have a question about um, sorption rates and how they might be potentially different between like eDNA versus pathogens. And then within those groups, of course, like the eDNA for like rare macroorganisms could be like in mitochondria, it could be in nucleuses, nuclei, it could be um, just naked DNA. And then the same it's like the same for pathogens, right? Like the nucleic acids are either naked or maybe they're stuck to like a particle or they're inside a capsid or inside a cell.
Yep.
Are you thinking that the form of the DNA or nucleic acids is going to affect the sorption rates? Are you seeing any evidence of that? And is it something you're going to look into?
So we haven't, in our experiments to date, we really haven't tried to parse those into different boxes. I think for sure their behavior would be different. Especially, you know, one of the things we're thinking about is like particle associated. You know, are you adsorbing viruses? Or are you adsorbing a particle that is a bus for a bunch of, you know, virions? Um, we don't have the data yet to kind of start teasing that apart, but my hypothesis is that it would certainly matter. What's interesting though is when we look in that wastewater data, despite all that, those kind of layers, we still kind of see this consistency across analytes. And so I think scientifically it's very interesting and would certainly you think matter, but maybe pragmatically in terms of quantification, maybe it doesn't.
Yeah, I mean,
Hard to say.
that's kind of weird. And I think all of us on this call who've been involved in wastewater surveillance in the beginning, we all were obsessed with all these like nuances and issues related to that. And in the end of the day, it kind of didn't matter. And it's like, well, that's weird. And what does that mean?
Yeah.
Yeah, and so I guess we're almost out of time, but I guess I was wondering, I'll ask a last question here is like, are you starting to think about like scenarios where passive sampling is needed versus not needed, maybe beyond the resource constraints, but like if you have a target that or a location where you're sampling with these characteristics, passive sampling would be more useful than actually grab sampling? Like, is there a like parameter space related to flow rate and number of people and concentration where you would expect these different methods to work better aside from like resource constraints?
Yeah, I definitely think that's in play. I mean, the challenge is that a lot of times in these studies, we haven't measured those parameters necessarily. And so it's hard to kind of take what you're observing and slice it up that way. But I definitely think, right? I mean, in terms of let's just pick prevalence, right? As a disease becomes more prevalent and people are shedding that material into your sewer, probably your heterogeneity between grab samples is going to change a bit. And maybe grab sampling becomes more reliable.
Mhm.
So I definitely think there's, you know, right tool for the problem that you're facing is definitely relevant. But I don't have the data yet to kind of
Mhm.
make recommendations.
It seems like partners would really like that kind of information so that they wouldn't have to wonder. Because I know we all get questions from our partners and municipalities and public health, and they often ask us, should we be using passive sampling? And it's like, it would be nice to be like, if the answer is yes to these questions, then yes.
Yeah, I think that's a great idea.
Yeah. Okay, well, we're out of time. So thank you, everybody. Thank you to Erin for the great talk and Fangqiong for her great talk. And we'll look forward to seeing you all in December. Happy Thanksgiving. Bye.